Okay, so I'll touch on crossbridge cycling. I got some questions on that. As a review, we'll go over the steps of that. And then we'll go over the steps of excitation, contraction, coupling. And then we'll look at some earlier stuff. Okay, so this is the schematic diagram of the cyclical process that is crossbridge cycling. Now, bear in mind, this is following the sliding filament model. And the sliding filament model is this theory that actin and myosin, which are the two filamentous proteins associated with muscle, they're going to be found in the sarcomere. They have to interact with one another. They have to slide across one another. And that's how we bring about shortening of the sarcomere. That's how we bring about shortening of muscle. That's how we bring about muscle contraction. So looking closer at the steps of this process, we'll start by looking at the first step, which is where myosin is in the cocked position. The myosin head is in the cocked position. And just to remind ourselves here, myosin has two binding sites, one to bind onto actin, one to bind that ATP to energize this whole process. So the first part, myosin is in its high energy state, right? It's loaded. It has that inorganic phosphate and that ADP bound to it. They have already been hydrolyzed. That was step 5. And we'll get back to that step as a part of this cycle. So as it's in that cocked position, that inorganic phosphate is going to be released off. And that's sort of the first part of this process. When that phosphate comes off, then we initiate the power stroke. So myosin is reaching up, it's grabbing onto actin. Now just to backtrack here, we need to have an understanding of the structure of actin. Actin is a fibrous protein. It's associated with two other regulatory proteins. One is called troponin, this small pink protein, and one is called tropomyosin, which is this thread-like protein that's wrapping around actin. Now, troponin has those 3 binding sites we talked about, one for calcium, one for actin, and one for tropomyosin. Calcium comes in, it binds, it uncovers those binding sites, and that's how myosin can interact, right? Myosin interacts with actin, that phosphate comes off, myosin drags actin towards the M line, which is the center of the sarcomere. Now, after the power stroke, after that dragging action, ADP is then released off. So ADP comes off, and myosin is bound to nothing. Nothing is bound to that ATP site on myosin. The next step is myosin is still tightly adhered to actin. What do we need in order to get myosin off of actin? What needs to happen to remove myosin from actin now? ATP, and new ATP has to bind to the ATPase site of myosin. This is why we have rigor mortis after death. So when someone dies, their muscles stay very tightly contracted because there's no ATP. There's no more cellular respiration, there's no more generation of ATP. And so if the muscles are contracted, they stay in that state, right? So a new ATP comes in, it binds to that myosin head, and then it can release off of actin, right, and go back into its resting position. So in this position, when ATP is bound to myosin, it's in its low energy form. It's not really attracted to actin, right? So it's going to come off, it's going to dissociate, and it's going to wait for that ATP to be hydrolyzed again. After that ATP is hydrolyzed again, the myosin head is now binding ADP and that inorganic phosphate again in that ready, loaded position, waiting for another opportunity to interact with actin. Okay, so it's going to go back to the high energy form, which is that cocked head of myosin. Okay, so these are the steps of crossbridge cycling. And again, this process is heavily dependent on the presence or absence of calcium. If there's calcium in the cytoplasm, we're going to have the uncovering of these myosin binding sites. These black dots are the myosin binding sites. If there is no calcium, what's going to happen? What's going to happen if there's no calcium in the cytoplasm? Perfect. Tropomyosin will not uncover those binding sites. So even if myosin is in its high energy form, it cannot interact with actin unless calcium initiates that process of uncovering those binding sites. Alrighty? Okay. All right, the next thing I'm going to review is excitation, contraction, coupling, and the steps of that process. And in that, we'll also look at what happens at the neuromuscular junction as a review of that as well. So we have a motor neuron, right? This is going to be an alpha motor neuron. It's innervating a piece of skeletal muscle, which is depicted here. And this motor neuron has an action potential running down this axon. When the action potential gets to the terminal bouton, what neurotransmitter is going to be released here at the neuromuscular junction? Acetylcholine, perfect. The only neurotransmitter at the neuromuscular junction is acetylcholine. Acetylcholine is released into the synaptic cleft. It crosses that synaptic cleft. It binds to what type of receptors on the motor end plate? So there are acetylcholine receptors, but we have two types. What type specifically is only found on skeletal muscle? Not adrenergic. Nicotinic, yes. So only nicotinic cholinergic receptors are found on the motor end plate. So acetylcholine binds to those nicotinic receptors. They allow the opening of channels. And so we depolarize the motor end plate. And what do we generate? What do we call the depolarization of the motor end plate? What do we call the depolarization of the motor end plate? An end plate potential. So we generate an end plate potential here. And that end plate potential, that depolarization, will then propagate down the length of the sarcolemma. And as we should remember at this point, the sarcolemma is nothing but the plasma membrane of muscle. Okay, so it's going to propagate down the plasma membrane, but then within the plasma membrane of muscle, there are these invaginations that we call T-tubules. And the function of T-tubules is to help that depolarization, that action potential, that end plate potential, propagate throughout the depth of the muscle. So it's like little tunnels that help that end plate potential to get deep within the muscle very quickly. And so now the action potential is propagating down the T-tubule, and it's going to get to a receptor in the membrane of that T-tubule, right? So there are two receptors here. There's a DHP receptor, dihydropyridine, which is in the membrane of the T-tubule itself. And then there's a ryanodine receptor, which is in the membrane of the sarcoplasm reticulum. Now, what's the function of the sarcoplasm reticulum? What do we say is the function of the sarcoplasm? No, sarcoplasm reticulum is in the cytoplasm, but it's not a part of the cytoplasm. What is it doing specifically? It's storing calcium. So the sarcoplasmic reticulum is the area where calcium is being stored until we need to release it into the cytoplasm. All right, so in this image, this blue area is representing the sarcoplasmic reticulum, whereas the white space is the sarcoplasm or the cytoplasm, right? And so what happens is when that voltage from the action potential gets to this DHP receptor, this is a voltage-gated receptor, it's going to send a signal to the ryanodine receptor because they're very tightly coupled. So they're touching one another. And so the DHP receptor passes the signal on to that ryanodine receptor. Now the ryanodine receptor is also a calcium channel, right? In it also being a calcium channel, it's going to open up once it gets that signal from DHP. When it opens up, it's going to allow calcium to move from where to where? Where are we moving calcium from at this point? Right, so we're going to move it from the sarcoplasmic reticulum where it's being stored out to meet the sarcomere, which is in, where's the sarcomere located? It's in the myofibril, which is in what space? The cytoplasm, right? So we're moving calcium from the SR into the cytoplasm, right? Now in this image, what we need to understand is that this space between here is what's not depicted over here, but it's important. This is how the calcium is getting out. So calcium is going from this blue area in the SR out through these receptors into this white space, which is the cytoplasm. And in this white space is where we have our myofibrils, which have our sarcomeres, which have the actin and myosin filaments. Okay, so now calcium gets out of the sarcoplasmic reticulum. It can easily flood the cytoplasm, and it's going to go and interact with actin molecules, right? So it interacts with actin specifically by binding where? Where does calcium bind here? We talked about this. Where does calcium bind? So calcium has one job in all of this. It's going to bind one place, troponin. Calcium binds to troponin, which is that pink regulatory protein here. And this is showing calcium in green. So they bind. And when that binds, the process we discussed earlier with tropomyosin uncovering those black binding sites, those black dots, and allowing the whole cycling of what we just discussed earlier to take place. When that is over, when all of that cross-bridging cycling is over, what needs to happen at this point to cease muscle contraction? What needs to happen at this point in order for muscle to cease contracting? Calcium has to leave the cytosol. It has to be removed. Otherwise, we're going to have those binding sites being continually exposed. Right? In order to stop muscle contracting, we need tropomyosin to cover those sites again. And in order to do that, calcium has to leave the cytosol and go back where? Where did it come from? The SR. Good. It has to go back into the SR. Okay, now we spoke about gradients, and now calcium naturally wants to go from the SR into the cytoplasm. So what kind of process is going to be required to get calcium out of the cytoplasm? What kind of process is it going to take to get calcium out of the cytoplasm and back into the SR? This is like a throwback question. It's going to require active transport, right? It needs to go against its gradient, so it's going to require active transport. And that's where the calcium ATPase pump comes in. So there's a pump on the membrane of the SR that is actively pumping calcium back into the SR, right? It has to go against its gradient here. So now it needs ATP. What kind of transport is this? Another throwback question. What kind of transport is this using ATP? So we know it's active. Is it primary or secondary? It's primary, right? Primary active transport, which uses ATP as the energy to drive those ions against their gradient. Don't forget that first initial stuff that we studied, all right? Remember your end of semester exam is comprehensive, okay? So that pumping of calcium back into the SR is what ends this entire process of muscle contraction and puts the muscle back at rest by covering up those binding sites once again. Okay. And here we can compare the tropomyosin and troponin in the relaxed state to tropomyosin and troponin while muscle is contracting.